students, welcome to Biomix Sub. In this today's video, we are going to discuss about the eye lens development in case of vertebrate. And this is a part of developmental biology. And from this topic, there are certain questions, experimental questions, which are asked in CSIR Life Science Net. It can be of 4 marks. So I hope after seeing this complete lecture, you can able to solve those questions. So let's get started with the video, guys. So what happens? There are different layers in the eyes. They are the retinal pigmented epithelium, the neural retina, and the lens. When the light falls in the eye, it is being transferred to the retina, and in this retina there are certain cells like rod cells and cone cells which absorbs this light and it responds and gives a signal to the brain. And this retinal pigmented epithelium and the neural retina which is present, they are being derived from central nervous system, and the lens which is present, it is being derived from non-neural ectoderm. Okay, so before going details, we will understand from where this ectoderm is being formed. So after the fertilization, there is formation of zygote. After the division of the zygote, after the developmental process, the zygote is converted into morula. After certain division, the morula is converted into blastula. After this, blastula is converted into gastrulation phase, and after the gastrulation phase comes the neurulation phase. Okay, in this gastrulation phase there are formation of 3 germ layers like ectoderm, mesoderm, and endoderm, and from these 3 germ layers there are formation of different parts of the body. And the lens which we are going to discuss, it is being derived from the ectoderm. And in the neurulation phase, there is formation of conversion of from the neural cell, there is formation of neural tube, and it is also made from the ectoderm. Okay, so details we will discuss in the going time. We will discuss how this process is being taking place. So for understanding the eye lens formation, we have to understand what is induction and what is competence. So what do you mean by induction? That means someone is inducing to another one. So some cell is inducing to another cell. Suppose this is cell A. This is cell A. This cell A is giving some paracrine factors, which is the transcription factor. So what do you mean by paracrine factors? Paracrine factors means these factors cannot be transported to long distance. It is a short distance transcription factor. If it is giving a signal, someone will receive this. So this is being received by cell B, and it should have a receptor with it to receive the signal. It is having a receptor, and after receiving the signal, the cell B will respond to it. So in this case, cell A is known as inducer, who is inducing cell B, and as cell B is responding to the signal, it is known as responder. Now the cell B is competent. Okay, so what do you mean by competent? Competent means the ability to receive a signal and respond to it. Okay, so this responder, now the cell B is now competent. That means it can now receive some other signal and it can also respond. So for example, we will understand, suppose this is a cell which is responsible for forming the lens. Okay, so but this cell requires some induction. It requires some of the transcription factors so that it will be able to synthesize the cell or produce the lens. Okay, so there are certain cells, like there are 2 signals required for this cell to become lens. So the first signal is given by cell A. The cell A will give one signal to this cell. Okay, so now after receiving the signal 1, this cell which is responsible for forming the lens, now it will be competent. Now it is competent, that means it is having the ability to respond to other signal, receive one signal and respond towards it. Now the cell B comes, the cell B which is present, it is the original cell which will give the signal to this to form the lens. So this signal can be received by this cell and it can respond it to form the lens. Now the lens is being formed. Okay, in this way the process is taking place. Okay, so already we have discussed what the 3 germ layers. They are the ectoderm, mesoderm, the 3 germ layers after the gastrulation phase. After the gastrulation phase, there are 3 germ layers: ectoderm, ectoderm, mesoderm, and endoderm. Okay, this ectoderm is having a place, it is having a particular place that it will make the lens, that is known as presumptive lens cell, presumptive lens cell. So let us see in the pictorial way. So here that in the mid gastrulation stage, what happens in the mid gastrulation stage, that is ectoderm and this is the endoderm. Okay, so this ectoderm which is present, it is having a particular place, it is having a particular place which is responsible for the formation of lens, and this is known as presumptive lens ectoderm. So what we have already discussed, that for the formation of lens it requires two signals. The first signal is that it will induce someone, and the second signal is the main signal. Now here this cell which is present, which is known as the presumptive lens ectoderm, first it needs to be induced by some cell. So the first induction is done by endoderm and mesoderm. You can see this arrows from here and one from here. They are inducing this presumptive lens ectoderm. So after the mid gastrulation stage comes the late gastrulation stage, and during this time, after this induction, there is after this induction there is formation of OTX2, which is being expressed by the presumptive lens ectoderm. Okay, now next comes the early neurulation stage. Okay, after this early neurulation stage, what happens, there is formation of PEX2. PEX2 is being expressed. So why this PEX2 is, how this PEX2 is formed? So the second, so here what happens, the first signal is being given by the endoderm and the mesoderm to the ectoderm. The second signal will be given by neural plate. So from where this neural plate is being formed, this neural plate which is present here, it is also from the anterior region of the ectoderm. So from this region, the neural plate is being formed, and this neural plate, it will give the second signaling to this presumptive lens cell, and after that there will be formation of PEX6. So in the early neurulation stage, what happens, there is formation of OTX2 and PEX6. Okay, now this PEX6, after the expression of the PEX6, what it does, it make competent to respond, it make competent to respond to optic vesicle. Okay, so from where this optic vesicle is formed, so here we can see this is the ectoderm, and below that there is the brain. Okay, here is the brain, and from this brain there is extension. In the two regions we can see there is extension from the brain, that is known as optic vesicle. And this optic vesicle, what it does, it will give the signal to the ectoderm. Okay, it will give this optical vesicle which is present, it will give a signal to ectoderm. Okay, how if this optic vesicle is giving some signal, how this cell is responding to it? It is responding because this cell is having the PEX6. What is this PEX6? This PEX6 which is present, it is giving the competency to this part. Okay, so what happens, this optic vesicle which is present, it will release some factors like FGF8 and BMP4. Okay, so how this is being regulated, how this is being regulated, let us understand here. So here there was formation of brain, and there was bulging from the two sides, there was bulging. We will discuss this part, how it is happening, how this is releasing some of the transcription factors. And after it was the ectoderm. Here there was formation of ectoderm is present. So from this, after getting the signal, here the optic vesicle which is present, it will give two signaling factors like BMP4 and FGF8. This BMP4 which is present, it will be transferred to the ectoderm. What it will do, it will be transferred to the ectoderm, and by responding it will give one, it will help in the formation of another one transcription factor which is known as SOX2. And the BMP4 which is present, it is a TGF beta factor. Okay, so responding to that BMP4, what it will give? It will give SOX2. From this also there can be a question that after the BMP4 what is formed when the BMP4 is being transferred to the ectoderm, what is formed in response? The answer will be SOX2. The next factor is a FGF8. After this FGF8 is transferred, it will follow the RTK pathway, and it will respond and it will give LMAF. LMAF is a transcription factor. Okay, so after giving this BMP4 and FGF8, this ectoderm is responding and giving the SOX2 and LMAF. Why it is giving this SOX2 and LMAF? Because this ectoderm which is present, now this ectoderm is competent. So why this is competent? Because it is having one factor which is known as PAX6. That is the reason it is competent. Now what happens? The SOX2, LMAF, PAX6 which is present, this three will help in lens differentiation. Okay, lens differentiation, and they will help in formation of lens. Okay, this is how the lens is being formed. Okay, so what happens in this case? PAX6, SOX2 and LMAF. What they will do? They will form another one factor which is known as delta crystalline, which will help in the production of lens. Now we can see here with this help of this diagram, SOX2 and PAX6 will help in formation of delta crystalline gene, which will help in the formation of lens. Okay, this is the condition how it is taking place. There is one of the experiment which we will discuss, like this things which are happening, they are happening in a particular place. Okay, there are some particular place where the eyes will be developed, like it will be in two sides of the brain. So if this is present, if the place is being changed, it cannot respond. Like we will see here, this is the head portion and the trunk portion. What happens in first case? Normal induction of lens by optic vesicle. Here there is normal induction of lens because this is a place where the optic vesicle is present, and this is a place where the presumptive lens formation part was present. So here there will be normal lens formation. Now in the second case, the optic vesicle cannot induce ectoderm, that is not competent. So this was the place of the ectoderm which was known as the presumptive lens cell, and this particular place was a place where the competence was formed, where the cell was competent. Okay, so the optic vesicle was giving the signal to it and the lens was formed. But here this ectoderm which is present, this ectoderm is not competent now. So here the optic vesicle which is present, it will not allow this ectoderm to form the lens. Okay, now the third condition, optic vesicle is removed, no lens is induced. If this cell is competent, but there is no optic vesicle present over here, in that case also here there will be no lens formation. Okay, in the fourth condition, what happens? Tissue other than optic vesicle is implanted, no induction occurs. If there are certain other tissues other than optic vesicle, it also cannot induce the cell for the formation of eye. Okay, this is how the eye lens development is taking place. So already we have discussed. So there are three steps of eye lens development. The first is the eye field and optical vesicle formation. Second is the lens placard formation. Third is the interaction between optic vesicle and lens placard. So first, what is happening? There is a particular place where the eye field needs to be identified, that in this region only there will be formation of eye field. So we can understand it with this help of this diagram. There is formation of noggin, and this noggin which is present, it will help in the formation, help in the secretion of the OTX2. This OTX2 in the stage 11, this noggin which is being induced, it is in the stage 10.5, and in this, after this, the OTX2 is been formed, and in the stage 11 there are other transcription factors also, other factors which are formed like ET, RX1, PAC6, 63, LHX2. What they will do, they will help in the specifying the eye field where the particular eye will be formed. Okay, so here after this stage like 12.5 stage, the ET, RX1, PAC6, 63, LHX2, T2, OPTX2, this all will be formed, and they will decide a place where the eye will be formed. So questions can be asked by giving this transcription factors or this parameters, that whether these are responsible for formation of the neural plate or not, or for the formation of the eye field or not. Okay. So now the next thing is the, let us see the mutation case. So this is a wild type mice which is present, and it is having normal eye. It is having the ability to form normal eye. Okay, but this is the PAC6 mutant. This PAC6 which was present, it was responsible for making the shell competent. That means it can respond to the optic vesicle and it will form the eye. But as this is mutant, in the surface ectoderm it should be present, but this is mutant in the surface ectoderm. That means it is not having the ability to form the eye. So let us see the experimental cases. Here the optic vesicle is present, here the surface ectoderm is present, and this is the lens induction. So here there is wild type, and the surface ectoderm is also wild type. That means there will be formation of lens. In the optic vesicle, there is PAC6 mutant. As we have already discussed, the PAC6 which is present, it should be present in the ectoderm, not in the optic vesicle. If it is present in the ectoderm, then it will help, that means the ectoderm is now competent and it will respond to optic vesicle for the formation of lens. But in this case, the PAC6 is mutant in case of optic vesicle, but the surface ectoderm is wild type. So in this case also there will be lens induction because the surface ectoderm is having the PAC6. Okay, now the third condition. This optic vesicle which is present, it is of wild type, but the surface ectoderm is PAC6 mutant. That means now the PAC6 mutant is present in the surface ectoderm. That means the surface ectoderm is not competent now, so it cannot respond to the signal which is being provided by the optic vesicle, and there will be no lens induction. Okay, now in the fourth case we will see the PAC6 is mutant in both the cases. So here there will be no lens formation because the surface ectoderm which is present, it should have the PAC6 to make it competent or to give the response or to receive the signal from the optic vesicle. Okay, so in this case in humans there is a condition which is known as aniridia condition due to the PAC6 deficient, in which the iris which is present, it is being disturbed or the lens is not formed normally. Here also we can see in case of cavefish, when it is normal, it is having the proper eye, but due to this cave dwelling population of this fish which is present, it is in this case the PAC6 is mutant and there is mutation of this eye. Okay, I hope you have understood this lecture and I hope you will solve the questions, and if you are having any doubt in the question, you can ask me in the comment section.